So thank you very much all for coming today to Jamie's end of PhD seminar. It's a delight to be able to introduce Jamie. So he started working with me in 2008 as an undergrad student. And when Doug first approached me and said, would you like to have this undergrad student in your lab? And he sent through the CV, I thought absolutely not. And not because the usual reasons of their track record wasn't good enough or anything like that. Jamie had done just maths and physics, and that didn't put me off either, but the fact that he'd only got 100% made me slightly concerned and thinking, oh, this is a lot of responsibility. But then getting over that and realising always hire people smarter than yourself, and then that was fine. And he had a wonderful undergraduate year, and that led on to a really great honours year where he topped the year. And during those 2 years, he came up with a list of epigenetic modifiers that we're now building shRNA libraries against. And so he's really enabled much of the research that's going on in my lab at the moment. And then during his PhD, he's used some of those libraries and has also developed some really great systems to be able to analyse X inactivation and screen for new modifiers of X inactivation. So hopefully you'll see that Jamie is not only remarkably talented in maths and physics, but also in biology. And so we really look forward to your seminar today, Jamie.
Thanks very much, Manny, for that lovely introduction. Okay, so today I'm going to talk about my PhD project entitled Screening for Epigenetic Regulators of X Chromosome Inactivation. So this is a little outline of the talk. First of all, I'm going to start with a very brief introduction on epigenetics and tell you about X inactivation as an example of a model system for studying epigenetic regulation. Then I'm going to introduce our shRNA library that we're using for screening, and then talk about a mouse embryonic fibroblast system that I used during my PhD for studying genes involved in the maintenance of X inactivation. And then one of the hits that came out of this screen, Setdb1, before I then talk about a novel screen that we're currently starting with embryonic stem cells. So first of all, epigenetics and X inactivation. So the common definition of epigenetics is mitotically heritable changes in gene expression without changes in the genome sequence. And this type of regulation essentially occurs at the level of the chromatin, whereby regions of the genome can be loosely packaged or tightly packaged, and this can influence the transcriptional state of the genome. And these different transcriptional states are associated with different histone modifications and DNA modifications. This type of regulation is really important throughout development for establishing and maintaining correct gene expression patterns. And although this is mitotically heritable, it also can be quite dynamic, so these patterns can be changed at different times through development, and can also go awry in disease. And one really striking example of epigenetic regulation is X chromosome inactivation. So X inactivation is a method of dosage compensation that exists in female mammals. So females have 2 X chromosomes and males have 1. So 2 copies of each X-linked gene in females and 1 in males. And to overcome this potential imbalance, one of the 2 X chromosomes in females is epigenetically silenced early during embryonic development. So in placental mammals, the choice of which chromosome to inactivate is random. So it can be either the maternal or the paternal X chromosome. And this is illustrated quite nicely with these mice with an X-linked heterozygous, this X-linked mutation that affects coat color. You can see 2 patches of fur corresponding to 2 potential choices of active X chromosome. And similarly here in these mice with a GP transgene, heterozygotes show this pattern of a mosaic pattern of expression due to random X inactivation. And also, then this sets up a situation in both sexes where there's only 1 X-link, 1 copy of each X-linked gene and 2 of autosomal genes. And this is less well understood, but there is actually, in many cases, upregulation of the X chromosome compared to autosomal genes. So in mice, this pattern of random X inactivation that you can see in adults is the second wave of X inactivation that occurs. So initially, there's an imprinted wave of X inactivation where the paternal X chromosome is specifically silenced from the 2 to 4 cell stage, and this silencing is complete by about the blastocyst stage. So this silencing is maintained in all the extra embryonic lineages. However, in the inner cell mass of the embryo, these cells reprogram and reactivate their inactive X, such they have 2 active X chromosomes. Subsequently, about day 5.5 of development, the cells of the epiblast then will undergo random X activation and independently choose to inactivate either the maternal or the paternal X chromosome, which gives rise to this mosaic pattern seen later on. So, as you might imagine, the inner cell mass of the blastocyst, which can be used to derive embryonic stem cells, is really useful system for studying X inactivation, because these ES cells derived from this region at this time point have 2 active X chromosomes, and they, in culture, then they will also undergo random X inactivation when they're induced to differentiate. So that's a really nice system for studying X inactivation in vitro. And also, I'm going to talk today about using mouse embryonic fibroblasts derived from day 13.5 embryos after X inactivation has been established. So we can use these sort of cells to study the maintenance and heritability of X inactivation. So I'm going to talk a little bit about some of what we know about the molecular mechanisms involved in X inactivation. And X inactivation is really controlled by this gene Xist, which makes a long non-coding RNA, and it's expressed from the inactive X chromosome. And there is a region on the X chromosome called the X inactivation center, and this contains a number of elements, including the antisense gene T6, which are involved in the repression of Xist expression in pluripotent cells. In addition to this, pluripotency factors such as OCT4 are able to either directly or indirectly repress Xist expression. It's then upon differentiation of these cells that they undergo random X inactivation. So what actually happens in these different early differentiating cells is the 2 X chromosomes initially repressing Xist expression co-localize in the X chromosome, in the nucleus rather. And there's a sort of crosstalk between these 2 X chromosomes such that factors are unevenly distributed on the X chromosomes, and one of them is protected from silencing, continuing to express the antisense gene T6, and the other one then upregulates Xist expression. And then this triggers, in the right context, random X inactivation, X inactivation of that chromosome. So Xist localization is controlled by initially YY1 and then HNRPU proteins. And you can see in this by RNA fish, initially you get 2 very small signals of RNA, Xist RNA expression, but upon differentiation, there's a rapid upregulation of Xist to form a repressive compartment, which is excluded for RNA polymerase II, and initially contains repetitive elements of the X chromosome. But then as differentiation proceeds, genes on the X chromosome are gradually recruited into this compartment, and the majority of the genes, in mice at least, are completely epigenetically silenced. So this repressive compartment also has a number of chromatin modifications associated with it, as it loses active marks such as histone, lysine acetylation, and transitions from euchromatin into heterochromatin. And so there are a number of other epigenetic marks, such as H3K9 methylation, that's enriched on the inactive X, and also H3K27 trimethylation, which is recruited by the PRC2 complex. So these are the initial and establishment phases of X inactivation, and X inactivation needs to be maintained throughout the life of the organism. So a number of other epigenetic regulatory events occur to keep this state stable. And interestingly, perhaps to this audience, is that deletion of Xist in female mice using a VAV-CRE actually causes hematological cancer after about a year or so latency. And although this hasn't really been characterized, essentially a large number of genes are dysregulated, as you might imagine, when you get reactivation of the inactive X, which causes a sort of myeloid proliferative disorder. Anyway, so a number of different things happen. The X chromosome becomes late replicating. A number of proteins, which of the function of which aren't quite clear, is recruited to the inactive X, like macro H2A. And one of the most important changes that occurs very late during this process is the DNA methylation of the promoters of X-linked genes in their CPG islands. And this is maintained by the DNA methyltransferase DNMT1. As you can see here, this lactate transgene initially shows random X inactivation pattern, but without DNMT1, a day later during development, you get reactivation, showing that without DNA methylation, this silencing can be rather unstable. SMOOCHD1 is also another protein that plays a role in the maintenance of X inactivation, although the mechanisms involved in this case aren't quite clear, but it is involved in also maintaining DNA methylation. So this is a summary of random X inactivation as it occurs during the differentiation of ES cells. And this sort of illustrates all the different epigenetic processes that are involved in X inactivation, which makes it a really nice model system for studying epigenetic regulation in general, as all these different processes are found in different epigenetic contexts. Even though it exists as specific for X inactivation, long non-coding RNAs are really important for other types of epigenetic regulation. So now I'm just going to introduce our shRNA library that I've been using for screening, with the idea that we want to find genes involved in X inactivation that we can also look up then in other epigenetic processes in our lab. So as Marnie mentioned, this is an shRNA library that we've made ourselves using a list of 1,100 or so candidate genes, which we've chosen based on the literature and based on known role in epigenetic regulation or potential roles in epigenetic regulation based on their domain structure or complexes they might form. So this is a focus library. So we're not looking genome-wide, but a lot of these proteins have not actually been characterized at all. So this is a good place to start for looking at epigenetic regulation. So we've designed 10 hairpins for each of our genes in this LNP-BFP vector context. So this is a retroviral vector which drives the expression of an artificial microRNA and has a puromycin-resistant gene and BFP as a reporter. And so this construct, when we infect our cells with a retrovirus, stably integrates into the genome and driving gene knockdown. So we divided our shRNA library into a few subsets, and the enzymatic DNA-binding helicase subsets, comprising about half the library, have been cloned now, so that we have about 5 hairpins per gene, and pretty much the majority of this work has been done by Tracy Wilson, doing a fantastic job with all this cloning. So the idea, we have lots of hairpins per gene. When we're using hairpins individually in individual assays, we test the knockdown for these hairpins, but if we're using them in a pooled approach, we don't validate them, but we've got lots of great hairpin depth, so we can feel confident that we have a good gene knockdown for at least 1 or 2 of these hairpins. This is just showing some qPCR data for some hairpins from our library, so often we get a lot of very nice hairpins. Sometimes we only get one or 2 that work well, but in general, we get quite good knockdown by the RNA level, and when we've tested the protein level, it tends to be a little bit better as well. So now I'm going to introduce the first screening system that I've been using, mouse embryonic fibroblasts. So in this case, using these MEFs to screen for genes involved in X inactivation, and using a X-linked GFP transgene as a reporter of X chromosome activity. So this transgene it consists of about 20 tandem copies of GFP on the X chromosome, and as you can see here, it's subject to random X inactivation and has been previously used in MEFs. So here's some flow cytometry showing in red here heterozygous females for this GFP transgene. They're either GFP negative or GFP positive, depending on which chromosome is active. So earlier during my PhD, I derived an immortalized cell line with the GFP on the Inactive X chromosome. Subsequently, I also used a slightly improved system using an allele of exist, which can't initiate X inactivation. So the idea of this is that we use this allele in combination with GFP because this one can't silence its chromosome. This then forces the chromosome with GFP to be the inactive X chromosome. So in this case, MEFs with this exist delta A allele and the GFP are completely GFP negative, and so we know that GFP is on the inactive X in these cells. And so from these sort of MEFs, I've derived a few other immortalized cell lines that I've also used in my assays. So the idea behind this screen is that we take MEFs with GFP on the inactive X, we infect with shRNA of interest, and then look for reactivation of GFP by flow cytometry. So X inactivation is very stable, and so it's only a sort of partial reactivation that we see. And in fact, we also have to treat with a sensitizing agent called 5-azacitidine, which interferes with the DNA methylation machinery, preventing the passage of DNA methylation patterns across cell division. So this means that DNA methylation is passively lost as cells divide, which destabilizes X inactivation and increases the background level of reactivation of the GFP that we can detect. So I've used these two sorts of cell lines and various different treatments with 5-azacitidine in these assays. And essentially I've started off with a individual screen using some validated hairpins, and then tried a pilot shRNA sequencing screen using a pool of hairpins, and then I've also looked at some hairpins from our shRNA library. So these are the results of the first individual shRNA screen, and you can see here showing relative GFP expression by flow cytometry compared to non-silencing hairpin. And so DNMT1 here in red is our positive control, which shows very nice reactivation of GFP compared to the non-silencing hairpin. So all these other hairpins that I've tested were known to be involved or associated with changes occurring at different stages of X inactivation. And what you can see here in magenta is that the two hairpins targeting Setdb1, which is an H3K9 Methyltransferase, a mark found on the inactive X, led to an increase in GFP expression. Because the background of this assay was quite variable, I then altered with the conditions a little bit just to clean it up a bit. So now we can see again that DNMT1 reads out quite nicely, as does Setdb1, and also this hairpin, which is our best hairpin targeting SmoochD1, now also reads out as we had expected. So this was nice proof of the assay could work to detect hairpins involved in X inactivation. So what we wanted to do next was to try and use a pooled approach to try and screen a large number of hairpins at once. So in this case, we make virus using a pool of shRNAs and infect our MEFs, and then take a DNA sample after selection to look at the relative abundance of all the hairpins in this population. Then we treat the cells with 5-azacitidine and then sort the cells now, the GFP positive cells and the GFP negative cells. Our hypothesis was that genes like DNMT1 leading to an increase in GFP when knocked down individually may be overrepresented in this GFP positive fraction compared to the GFP negative fraction. So for each of these DNA taken from these different samples, we amplify the shRNA integrants and with unique reverse primers so we can pull them together and then sequence them. So initially it was about 140 hairpins, including negative and positive controls in biological triplicates. And this analysis of this particular experiment was done by Matt Ritchie. What was apparent was that this assay was actually quite variable. So you can see here that the three initial samples cluster very well together, but once we add 5-azacitidine into to the cells, things become a lot more variable due to the sort of stochastic nature of how 5-azacitidine works, probably reactivating different portions of the X chromosome in different cells. And we know that when we knock down DNMT1, we don't get 100% reactivation. It's only about 30% reactivation. So the changes weren't entirely consistent between the different replicates. Nevertheless, we were pleased to see that the top hairpin overrepresented in the reactivating cells was a hairpin targeting Setdb1, which validated the individual assay. And another hairpin a little bit further down, PRDM 2.7, also read out individually, and I'll talk about that a little bit later. But you can see that very few changes were actually significant due to this variability. And our positive controls didn't behave quite as expected either. So DNMT1, although that read out how we expected, SMUCHY1 actually went in the opposite direction. So things weren't quite working as how we had hoped. So we tried to optimize this a little bit using some different cells and varying the 5-azacitidine treatment. But this led to similar problems, essentially. So you can see here log-fold changes for all the hairpins in our pool. And this hairpin here is DNMT1, and this hairpin, next hairpin down is false positive. So there's there was very little separation between the signal in this and the background. So we've decided that pooling shRNAs and screening them in these particular MEFs with this assay is probably not the most efficient way to screen, and I'll talk about a much improved screening system in the last section of my talk. However, we were able to pull out Setdb1 as a gene implicated in the maintenance of X inactivation using both the individual and pooled screens. So we then wanted to go on and try and validate Setdb1's role in X inactivation. So as I mentioned, Setdb1 is an H3K9 methyltransferase. It's not been shown previously to have a role in X activation, but we know that H3K9 methylation is a mark found on the inactive X chromosome. And Setdb1 is quite important developmentally. Knockout mice die about day 7.5, and you can see the embryonic part of the embryo is severely deformed. Setdb1 plays an important role in ES cell identity, and it is involved binding with OCT4 to suppress genes involved in trophectoderm, trophectoderm like CDX2. So interestingly, Setdb1 knock down ES cells when they're injected into early 4 to 8 cell stage embryos will actually incorporate into trophectoderm when they shouldn't. So indicating that they play an important role in keeping ES cells Setdb1 also plays a role in endogenous retrovirus silencing and transgene silencing, and it's been shown to be an oncogene and amplified in lung cancer and melanoma. So the first thing we did to try and validate Setdb1 was to turn back to our shRNA library. And we knew that H3K9 methylation was on the inactive X chromosome, so we wanted to look at other H3K9 methyltransferases, which all share a common set domain, and see whether they could lead to a similar level of reactivation in our MEF system. Only one of these proteins, EHMT2, has previously been looked at in X inactivation, but didn't show an X inactivation phenotype in embryos. And PRDM2 and Sub39H1 and H2 double knockouts can survive till adulthood, suggesting that they don't play a critical role in X inactivation, because we'd expect at least female-specific lethality. However, we tested all these hairpins in our MEF assay. So you can see, again, this is non-silencing control hairpins, and the two Setdb1 hairpins from before show a nice GP reactivation, as do other hairpins from our shRNA library. All of the other hairpins that we tested targeting these other H3K9 methyltransferases, and at least 2 of these showed similar level of knockdown to the Setdb1 hairpins, so about 40% of wild-type levels. They didn't show the same sort of phenotype, suggesting that Setdb1 is most likely the methyltransferase involved in maintenance of X inactivation. The exception here was this PLEM2 hairpin, which is the same hairpin that came out of our pooled shRNA screen. But when we look at the relative expression of Setdb1 and PLEM2 from the RNA from these cells, you can see that Setdb1 knockdown kind of correlates with GFP activation, whereas the PLEM2 hairpin doesn't actually, that reads out, doesn't give a knockdown any different to any of the other hairpins. So this is most likely an off-target effect. And so by having multiple hairpins, this helps to control for these potential off-target effects. And this is just to show, again, that using the hairpins from the library in some slightly different maths, using different conditions, confirming Setdb1 as involved in maintaining X inactivation. So we wanted to then look, try and confirm these results using a system moving away from knockdown. And we were very fortunate that Catherine Carmichael in Ben Carl's lab had a Setdb1 gene trap, which she's kindly let us use. So this gene trap has beta-galactosidase neomycin fusion gene integrated into Setdb1, which produces this fusion transcript that lacks all the functional domains of Setdb1. And as with the published knockouts, the heterozygotes are fine, but the homozygotes die about day 7.5. So we wanted to examine X inactivation in these Setdb1 gene trap homozygote embryos. So to do this, we then again used our exist delta A allele and a GFP transgene. And then we wanted to look at this exist delta A GFP embryos, so embryos which should have forced the X inactivation of GFP, and look at homozygotes, heterozygotes, and the wild-type embryos to see what effect Setdb1 might be having on maintaining this silencing in vivo. So this has previously been used to look at other genes in X inactivation, and this is just an example of SMUHD1. So SMUHD1 wild-type and heterozygous embryos with this exist knockout GFP, GFP negative, whereas SMUHD1 homozygote embryos now reactivate their GFP. So SMUHD1 is important in maintaining X inactivation in these embryos. So we saw something similar in our Setdb1 gene trap homozygotes too. So this is an embryo, a wild-type embryo without the exist knockout. You can see that it expresses GFP in the embryonic part, showing random X activation. When we add in the exist knockout with the GFP, now they're completely GFP negative. However, in our Setdb1 gene trap homozygous embryos, we can see GFP expression throughout the embryo. So these embryos are just like the knockouts, severely deformed at this time point, but they we can see GFP suggesting that Setdb1 is keeping the inactive X silent. So I've shown you so far that we have shown Setdb1 having a role in maintaining X activation in vitro and in vivo using this GFP transgene. Next, we wanted to look more at endogenous genes on the X chromosome to see if we could confirm this result, and then also look at some of the, see if we can find Setdb1 binding to and look at H3K9 methylation on the inactive X chromosome. So to do these next couple of things, we have used another strain of mouse called the PGK-X strain. So this strain has its X chromosome derived from a wild strain of mouse, PGK musculus, and this has been back-crossed onto Black6, which is an inbred strain of mouse, such that all the autosomes are of inbred origin, but the majority of the X chromosome is from this wild background. This means that there are lots of SNPs that occur in genes and express SNPs as well through much of the X chromosome. This means that now we can have a transcriptional readout of multiple genes on the X chromosome. So what we've done, again, we've taken the exist delta A, crossed that to PGK, And we know then that this forces non-random X inactivation of the PGK chromosome. So in these cells, we expect to see only RNA from the inbred chromosome. And then by RNA sequencing, we can look for re-expression of the PGK allele when we knock down Setdb1. And so the importance of this system really is that using these SNPs, we can detect small, subtle changes in the level of transcripts that may not be detected if it's only, say, 1 or 2% of cells reactivating their inactive X. We'd be able to detect that by RNA sequencing, which might not necessarily be reflected in the change in the total level of RNA for a given gene. So we wanted to do this in vitro, and we decided to combine our Setdb1 gene trap allele with Setdb1 knockdown and make MEFs, and to try and get the lowest level of Setdb1 expression that we could. And so comparing then Setdb1 gene trap heterozygote MEFs back to wild type MEFs, with these ones with the Setdb1 knockdown and these with a non-silencing control. So then we've taken these MEFs, we've harvested RNA to look for the presence of PGK transcripts in the Setdb1 depleted cells. And we also combined with or without 5-azacitidine treatment to see if this made any difference to reactivation. Finally, we also sequenced a PGK homozygous control. So this is to enable us to identify where the SNPs occurred on the PGK chromosome. So we obtained a large number of reads for all of our samples, particularly the PGK control. And this analysis was done by Matt Ritchie and Catherine Phillips in his lab. We were able to identify 270 SNPs in about 70 genes, which were different in the PGK on PGK chromosome. So you can see in the PGK control, expression of the PGK allele is very high, but the inbred reference allele is not expressed. In the control sample, so the non-silencing hairpins, we get expression of the reference allele, but not the PGK allele. So then we looked at in our Setdb1 knockdown cells to see whether we could find increased expression of PGK. But we didn't see a widespread reactivation across the inactive X as we'd hoped. And we only found 6 SNPs in 5 genes that showed higher PGK counts in Setdb1 knockdowns compared to the control samples. And only one of these genes, RPS4X, was actually highly significant. So you can see about 1% of the reads in this case are from the PGK-X chromosome. So this perhaps suggests that Setdb1 is may not be involved in silencing all of the X chromosome, or perhaps the level of sequencing depth that we had was not sufficient, even though it's quite deep. It wasn't sufficient to detect really subtle changes of only 1% of reactivation that we might see at other genes. So this was a little bit disappointing, but encouraging that at least we can see one gene behaving as we had expected. So now we're going on to use these MEFs to look at the chromatin of the inactive X chromosome. And the idea behind this is to use Setdb1 chip as well as H3K9 di- and trimethylation chip to see whether we can detect these modifications on the inactive X. And as before, we can use the PGK SNPs to map our reads to either the active X chromosome or the inactive X chromosome. So we're doing that at the moment in collaboration with Andrew Kennery in our lab. And we have some preliminary chip PCR data for Setdb1 showing we can pull down Setdb1. So not at beta-actin, but at these 2 ES cell genes bound by Setdb1 in ES cells, which is encouraging. But also, you can see here 2 X-linked genes, CHIK1 and G6PDX, and they've been previously shown to be marked by H3K9 dimethylation in this paper here in female cells, but not in males. So this is an H3K9 methylation on the Inactive X. And we can see Setdb1 enrichment at G6PDX. So perhaps this is an indication of Setdb1 binding on the Inactive X. And we're going on to doing our chip sequencing sample to look at Setdb1 across the X chromosome. So finally, I'd like to talk about a novel screening system that we've established in the lab now using embryonic stem cells, using 2 different fluorescent reporters to monitor X chromosome activity. So these alleles were designed by, I can conceive and designed by Marnie as a way we can look at both X chromosomes at the same time. So these are knock-ins of either mCherry or EGFP at the 3' UTR of HPRT. HPRT is an X-linked housekeeping gene, so expressed at a constant level, and it's subject to random X inactivation. And so we've got HPRT, an internal ribosomal entry site, and then our fluorochrome. So this is translated separately from HPRT. So these are some ES cells with the GFP and cherry knock-in alleles on the different X chromosomes, and you can see that they are double positive, so expressing cherry and GFP because they have 2 active X chromosomes. And you can detect it easily by flow cytometry. You can also see it by microscopy, although it's pretty weak. So initially we were thinking about doing an imaging screen, but then we moved to using flow cytometry instead. And then you can see as ES cells differentiate, they become single positive. And MEFs have undergone random X inactivation, so they are either GFP or cherry positive. So these red and green ES cells are a screening system. We've decided to call them Christmas ES cells. And I came up with the most tortured acronym I could possibly think of to involve a color that was red and green to make X-linked Mazid and SMRAG stand for Christmas. And then Marnie came along and then suggested X-linked markers active silent, which is slightly more practical. So that's what we've gone with. Yeah. Anyway. So we've derived a lot of these ESL lines now in low oxygen, feeder-free conditions and a serum-defined, serum-free defined medium with these 2 inhibitors targeting GSK3 beta and MEK, and also with LIF. So these are the best ESLs that we can have to try and keep these cells pluripotent and having 2 active X chromosomes. So then the idea behind the sort of screening is that we take these ES cells, infect them with shRNAs from our library, select for infected cells, and then differentiate them by removing these inhibitors and lift, and then monitoring random X activation. So depending on which hairpins we use, we might expect to see a delay or a failure X activation, or perhaps reactivation of the Inactive X over the course of differentiation. And all this establishing of this ESL screening system has been done with Andrew Kennery. So first of all, this is showing by flow cytometry the differentiation kinetics of our ES cells. So because the ES cells don't really like to have their inhibitors removed, we gradually transition them into normal DME plus FCS. And then we've imaged them every day and looked at their expression of GFP and cherry. So what you can see is for the first 5 days of differentiation, they maintain roughly the same sort of level of double positivity. But then by day 6, day 7, they rapidly become single positive, either cherry or GFP positive, as HPRT is silenced and a GFP or cherry expression is lost. So this is nicely consistent between different ES cell lines. So then we went on to perform a little pilot screen in ES cells where we've infected with a few controls and also Setdb1 to see what effect that knockdown would have on our ES cells. And then we've expanded these cells then before we differentiated them and monitoring by flow cytometry the expression of both cherry and GFP. So first of all, this is showing our uninfected and luciferase, luciferase negative control hairpin, showing the same kinetics as before. And now also DNMT1 and SMUG1 hairpins, which are involved in the maintenance of X inactivation, don't show an effect. So we were thinking perhaps we would get reactivation at later stages of the inactive X with these knockdowns, but we weren't able to successfully culture these ES cells yet much past this point. So perhaps if we can extend the culture period, we may see these cells reactivate their inactive X, but we're not sure. However, the first thing we did see was that both the Setdb1 and HNRNPU knockdown ES cells prematurely differentiated. So you can see even before we've begun to remove the inhibitors and LIF, these cells are becoming single positive and become so quicker than the negative controls. So this makes sense from what I told you about Setdb1's role in maintaining ES cell identity. And it also turns out that HNRNPU, which was included as a control for the establishment of X activation, as it's involved in exist localization later on, it actually activates the gene encoding OCT4, which is an important pluripotency gene. So this explains why this gene was also dropped out early. However, we were very encouraged to see that two of our knockdowns, YY1, which is involved in the very early stages of X inactivation and exist localization to the X inactivation center, failed to properly inactivate its X chromosomes, as did JARED2, which is a PRC2 accessory factor, which is, has a role in recruiting PRC2 to the X chromosome. So this was nice that we were able to get our assay to read out. But we still thought we'd like to have a look at Setdb1 in this assay, but try and get around the ES cell phenotype that it shows by infecting the cells during our differentiation. So we infected our cells 2 days after we had begun the differentiation protocol. And now what you can see is that we've got uninfected and non-silencing hairpins behaving as expected, showing the same kinetics as before. But now both Setdb1 doesn't drop out and behaves exactly the same as the negative controls. YY1, which previously showed a failure of X activation, now no longer shows any difference. But HNRNPU, which previously dropped out as involved in ES cell identity, is now showing an X inactivation phenotype. So what we think is happening in these cells is that by infecting them quite early, we were able to get sufficient knockdown to look at X inactivation initiation events and also establishment. However, we were also picked up ES cell identity phenotypes. Then by infecting later, we didn't get knockdown early enough to pick up these very initial events of X inactivation, but now establishment events were, in terms of HNRPU, was uncovered because we lost this ES cell effect. And Setdb1 again then showed no phenotype, suggesting that it's probably not involved in the early stages of establishment of X inactivation. And then together with our MEF results, this suggests that it's probably more likely to be involved in the maintenance of X chromosome activation. So finally, we are now trying to expand this to use a pooled screening approach with our ES cells, using about 1,000 hairpins from our enzymatic subset of our library with appropriate ES cell controls. And the idea here is that we're going to monitor representation of hairpins over time, but as we differentiate our cells, but also looking at the double positive and single positive populations and comparing them. So we might expect that YY1 is more likely to be found in the double positive fraction compared to the single positive fraction based on the individual screening results. And we're also now going to be doing early infections and late infections to help us tease apart initiation, establishment, and any ESL phenotypes that we see from this screening approach. So finally, just to summarize, I showed you today that we've found that Setdb1 has a role in maintenance of X inactivation in vitro and in vivo using this GP transgene. Some evidence for reactivation of endogenous genes, and we're now starting to do CHIP to look at Setdb1 actually on the inactive X chromosome and determine whether it's actually laying down H3K9 methylation as we would expect. And our ESL results have shown that Setdb1 confirms the identity phenotype, but shows that it doesn't have a role at the very early stages of X inactivation in this system. And our shRNA library has been a really brilliant resource, not just in this project, but in other projects in the lab, and particularly for following up hits like taking out the genes, or the other H3K9 Methyltransferases to follow that up. MEF screening was able to identify Setdb1, although the pooled screening approach wasn't quite so successful. But now we've got this really novel screening system in ES cells to look at the early stages of X inactivation. We're going on now to screen in a pooled way in this system, hoping to pull out some more hits from our library. So finally, thank you to my supervisors, Principal Supervisor Marnie Blewett, who's been a really fantastic supervisor, and she's been a great help, and not just during the PhD, but during honours, and ever since I've been in her lab, it's been a real privilege to work for her. And also to Doug Hilton for letting me join his lab in the first place, and also letting me stay for quite such a long time. I thought he'd probably have found me out by now, but I've got this far, so that's good. Miha in the lab does all the genotyping and is really helpful as a research technician doing cloning and that sort of stuff, helping out with all sorts of experimental techniques. Joy, a former student in the lab, was involved in some of the later MEF pooled screening approaches. She did a lot of work on that. With Andrew Kennery, I've been doing all the ESL screen establishment and some of the ChIP-seq stuff, and it's been really great collaborating with him. Other members of the lab who have been great to work with. Christine helped with some of the E7.5 embryo dissections. Matt Ritchie is really helpful with analyzing shRNA sequencing data, and with Catherine also RNA sequencing data. Thanks to Gordon Smythe for bioinformatics discussion, and he's also on my PhD committee, as is James Murphy and Kurt Lakovich, and he, along with Kelly Rogers, were helpful for imaging stuff, and when we thought we were going to be using imaging screen for our ES cells. And Anna Voss is the chair of my PhD committee, and thank you to her and to Natalie from her lab for ES cell culture protocols. Pretty much all of this work wouldn't be possible without Tracy Wilson, who cloned pretty much the entire library, thousands and thousands of hairpins. She's done a fantastic job. Gianni was involved in the design of the shRNAs, and Adrian also involved in the early stages of the library. Thank you to Catherine Carmichael and Ben Kyle for the Setdb1 gene traps, and Graham Kay, Marnie's collaborator in Queensland, for the existed Delta A mice and the PGK strain. And also thank you to all the mouse people, particularly currently Ketty and in Kew here in Parkville, and Melanie in Bundura, and all the past mouse techs as well, for all the fantastic work they do helping us out with our mice. Thank you very much.
Okay, questions? Yep.
Great talk, Jamie. I was interested if you looked at your Xmas alleles and see how they behave in vivo during earlier embryogenesis.
So we have had a little look in vivo. We did try to look early on at blastocysts, but at that stage, because imprinted X inactivation is not fully complete, all the cells of the embryo were expressing both fluorochromes. So we weren't really able to image early dynamics in vivo as we'd hoped. We also had a look at LSKs in Christmas mice with the thought that perhaps as a stem cell population, maybe X activation may be slightly destabilized in that context. And also we thought that if we could detect reactivation of the inactive X in these cells, we would get a Nature paper pretty quickly, but we didn't see that, so it was fun to look anyway.
In your pooled shRNA screens, different cells, even though they're homogeneous, may expand at different rates, and that may therefore miss any differences in effects of each of the shRNAs. My question is, how many cells per transgene were actually in the cells in culture that you then assayed for?
How many happen cells per happen?
Correct.
Probably at least, I think when we sort of sequenced the cells, I think we aimed something like 10,000 cells per happen, which is a kind of what we found is kind of a good estimate. What I think the problem was in part with that assay was that at the later stages, because of, so there were two problems. Either initially the 5-azacitidine is quite toxic, so it kills a lot of the cells, or later on we tried to vary the 5-azacitidine concentration to reduce this background level. That also restricted the number of cells we got out the end. So either way, although we put a lot of hairpins in, we're only sampling a small number of hairpins, a small number of cells at the end, which is kind of the sort of sampling error, if you like. So this is probably kind of what makes it so variable at the very end, when we were dealing with less than 500,000 cells in the end from the GFP positive fraction.